In this lecture, we're going to take a closer look at the generation of bioelectrical energy, and we begin by examining this concept or this observation of what's called the resting membrane potential. And what that basically means is that there is an electrical charge separation across a membrane, so between the inside of the neuron and the outside of the neuron at rest. Now, at rest, a neuron is not triggered to release a neurotransmitter if that neuron has not been activated. Looking at another cell, a muscle cell, for example, the function of a neuron is to communicate from the one cell to other cells. The function of a muscle cell is to produce force. So muscle cells get activated the same way neurons get activated, except that the final output of a neuron is to release a neurotransmitter. The final output of a muscle cell is to produce force. But at a resting state, neither one of those are happening. The resting membrane potential refers to that electrical charge separation between the inside and the outside of the cell. And depending upon the type of cell, there is a range of that resting electrical energy difference. And it ranges anywhere from -80 to -60, depending upon the cell. So what this says is that the inside of the cell has more of a negative electrical charge with respect to the outside of the cell. And because of that negative inside, we say the cell is polarized. So when we think about polarized, a pole, positive pole, negative pole, if the cell is not neutral electrically inside and it is not under resting condition, we say that it's in a polarized state. And that refers to the electrical state of the cytosol, which is the interior fluid of the cell, versus the extracellular fluid. Now, if we take a polarized cell and we depolarize it, then the cell is no longer in a polarized state. So how do we get a neuron that at rest is polarized to a state where it's no longer polarized in this exact manner? Well, the easiest way to do that, if we take a look at the negative values, is to add positive values to it. So we can just do the math and add positive numbers to -80 or -60. And as you do that, -60 becomes -59, -58, -57, and it keeps moving towards the zero and maybe even into the positive direction. So how do we do that biologically? If we add ions that have a net positive charge to the inside, which is already polarized at -60, adding more positive ions will move the electrical environment inside the cell, which is polarized at -60. And for every sodium ion that we add in, that interior electrical energy state with respect to the outside becomes -59, -58, -57, 56, so on and so forth. Now, we can depolarize a cell by doing this. After a cell is depolarized, can we send it back to its polarized state? So after a cell has been depolarized, can we repolarize that cell? And yes, we can. So we're asking here is, can we restore the cell to its polarized state? That's what repolarization is. If you take a polarized cell and make it even more polarized, so if that resting cell is -60 inside the cell with respect to the outside, and you make it even more polarized or hyperpolarize it, it becomes more like -70, -80, maybe -85. So that's what hyperpolarization does. And there are two types of biological signals that can do this. Those are an electrical input to that cell, or a chemical input to that resting cell to change it. And to better understand this, we take a look at the concentration of some of the major ions inside the cell versus the extracellular medium. And we've got two different types of cells here. Here we have a squid neuron, and at the bottom part of this table is the mammalian neuron, or a human neuron here. Now, the reason we look at the squid neuron is because many, many decades ago, before technology allowed better resolution and magnification in studying how neurons operate, neuroscientists often used neurons harvested from the giant squid. These are exceptionally large neurons, uh, especially those motor neurons, which can be viewed with the naked eye. And so, with the magnification power at the time in the 1950s, they were able to generate quite quality images, but they needed a very large structure to do that. And that's why they used the squid neuron. But the major ions I'm going to focus on is potassium and sodium. And when we take a look at the mammalian neuron with potassium and sodium, the absolute numbers are quite different. We can see that. But what is quite similar between squid and mammalian neurons is the ratio of these ions with respect to the inside and outside of the cell. So we see with potassium in the squid neuron, much more, at least one order of magnitude, more potassium inside the cell than outside the cell. Sodium is opposite. One order of magnitude more sodium outside the cell than inside the cell. And we see the same pattern emerge for the mammalian neuron. So the mammalian neuron, an order of magnitude greater concentration of potassium inside the cell. The reverse here for sodium, which exists in much greater concentrations outside of the cell. And these are the concentrations in the resting cell. The resting cell. And so the action potential is the fundamental physiological event that gives rise to a bioelectrical signal. So whether it's a muscle cell or a neuron, the cell membrane is responding to what are called graded potentials. Potential is a catch-all term which refers to the potential energy, the electrical energy change that occurs between the inside and the outside of the cell. And so in a polarized cell, remember, when the cell is resting, it's in a polarized state. A depolarizing current will achieve a critical threshold. So the polarized state of -60 moves a little bit below -60, a little bit above -60. But if there's a signal that's going to try to move the -60 into a depolarization direction, there's a critical threshold that would cause the cell to fully depolarize. And we'll see that here in the next on the next slide. When we talk about current, current is like flowing water. Current is the flow of ions into the cell, whereas voltage is the energy difference from one side to the other side. So the voltage drives the current. For water to flow along a river, there has to be a height differential or some sort of turbine that pushes the water. The turbine is the energy, the flow of the water is the current. So, how do ions flow into the cell? Remember, at rest, more sodium ions outside the cell, sodium channels have to open. Now, what will cause a sodium channel to open? It will be a chemical or an electrical stimulus. If we're talking about stretch receptors, it could be a mechanical stimulus. But in our example here, we're going to focus on a chemical stimulus because the chemical is the neurotransmitter that would open up sodium channels on the dendrites of the subsequent neuron. So with an action potential, what happens is that there's a large deflection in the membrane potential, so that electrical state of the membrane changes from negative to positive. So remember, the cell at rest is polarized at about -60 or -70, again, depending upon the cell. So in this example, if we have a particular cell that's resting at -70 millivolts inside with respect to the outside, an action potential will cause that interior environment to move to +30. Our polarized cell has now depolarized to +30. This is a very fast event. In neurons and skeletal muscle cells, remember, this is an event that is confined to the membrane of these cells. The event is very fast. It begins and ends within 1 to 2 milliseconds. But not all cells respond the same way. Most of the cells in the mammalian heart or human heart, most of the cells have very long events, very long action potentials, on the order of up to half a second. And those are cardiac muscle cells. So on this graph, we're going to plot the membrane, the typical action potential of a neuron. And as we can see along the y-axis, what we're plotting is that energy, electrical energy separation between the inside of the cell and the outside of the cell. So our resting membrane potential in that resting neuron, it's not releasing a neurotransmitter, is -60 millivolts. And there's our units up on top, millivolts. Now, suppose we apply a stimulus to that particular neuron. And then we apply another one, and we keep doing this. And what we notice here is that the stimuli to the neuron, this could be a chemical stimulus or it could be an electrical stimulus, but these stimuli are closer. So the frequency of the stimuli to the neuron is increasing. It's getting faster. Now, what effect does that have on the membrane state and the internal-external state? So what you see here is that as you apply a stimulus, actually this arrow should be over to the left a little bit more, as you apply this stimulus, you see that the -60 starts to depolarize. So we're looking at -59, -58, -57, -56, and then it returns back to the baseline. So this here is not the action potential. This is what's called a depolarizing potential. It's trying to, it actually does cause some depolarization, but not a lot. And the cell returns back to its resting state. So what we do is we hit it again, and it will start to depolarize again, and it starts to rest. But before it gets back to the base electrical baseline, we hit it with another stimulus. And so these are what are called graded. These are graded potentials here. As we add another stimulus before complete membrane restoration, the next depolarization climbs up a little bit more because the cell has not fully reestablished the position of all those ions, but we can still activate it again. And we do that repeatedly before it can get a rest, before it gets to the resting state, and it slowly starts to decay. It decays towards what's called a threshold. And there's a critical threshold that when that neuron hits it, we see a very fast, almost impactful or violent change in its polarized state into a positive direction. Remember, this is inside the cell at +30. At peak here, at the peak point here, that's called the depolarized state. The event from threshold up to the peak is called depolarization. So this is depolarization. This is the fully depolarized state, and the decay from the baseline is being driven by sodium channels. They're being driven by sodium channels. So sodium channels are opening, and then many sodium channels open at threshold, and we see a massive rush of sodium ions inside the cell. But by the time the membrane or the cell reaches fully depolarized status, the sodium channels have actually closed at the peak point. So sodium channels are opening, they're opening, they're opening. Now they start to close, and they're fully closed at the depolarized state. And at the fully closed state, sodium ions are no longer moving. Potassium channels, which are embedded in the membrane, will start to open at the depolarized state. And when potassium channels open, as we added lots of sodium to the interior of the cell, potassium will start to escape the cell. Remember, more potassium inside than outside. And that will have the effect of repolarizing. So we're polarized, we've depolarized, and we're repolarizing, and that's driven by potassium leaving the cell because the depolarized state is going to open voltage-gated potassium channels. And the cell returns back to its baseline. However, it not just achieves the baseline, but it undershoots it as well. That's called a hyperpolarized state. So not only is the cell polarized once again, it's even more polarized. And what's the functional implication of the action potential, the full action potential of a neuron, is that during what's called the absolute refractory period. So the absolute refractory period is measured from the start of depolarization at the threshold to where the membrane or the cell has repolarized below the threshold. During that period of time, the cell cannot generate another action potential during that time. It can do that if it's below the threshold, but if it's achieved threshold, the absolute refractory period, that cell cannot generate another action potential. Now, after the cell has moved back towards repolarization and has come under the threshold, can we actually generate another action potential in that cell? It is possible because we enter what's called the relative refractory period. So during the relative refractory period, the cell can generate another one of these events, but the membrane and the cell has not completely restored back to its resting state. So it's going to take a stronger stimulus in order to generate another action potential in that cell. So this ends our video here on the action potential.